the Team 4 biology teacher, and today we're going to review DNA structure and replication. Now DNA, which is deoxyribonucleic acid, is a nucleic acid. It's one of the 4 macromolecules needed for life. And because it's a nucleic acid, the monomer that makes it up is a nucleotide. And this is a picture right here of a basic shape of a nucleotide. They're made of this phosphate group, this circle, a deoxyribose sugar, which is this pentagon, and a base, this rectangle right here. Now the base is how you identify the nucleotide. You should know these 4 bases: A for adenine, G for guanine, C is cytosine, and T is thymine. Now DNA has a pretty complex structure. So you see this one nucleotide right here, I have it circled in the picture. These nucleotides are linked together through the sugar and the phosphates. The bond that holds them together is a covalent bond. And so you can see them here on the left as well as here on the right. You have covalent bonds. That makes up the backbone of DNA. So we say it's a sugar-phosphate backbone. Now in the center, you're going to have your nitrogen bases. Your nitrogen bases face each other and they are complementary. So C and G are always going to pair together, and A and T are always going to pair together. And the way to remember that, apples are in the tree, cars go in the garage. So A with T, C with G. And it's very possible on the air test they would give you a sequence that's one side of DNA and ask you to write out the complementary sequence. So C would go with a G, A with a T, G again with C, T with an A. Now in between the nitrogen base pairs, it's important to understand that we are linking them with hydrogen bonds instead of with covalent bonds. And that's simply because they are weaker bonds and it's necessary to allow DNA to be broken. On top of this, DNA is considered anti-parallel, meaning in one side it goes from 5 to 3 prime, and on the other side it goes 3 to 5 prime. They go in opposite directions, either side of the DNA molecule. You can kind of tell that easily by looking at the orientation of this sugar. The one side has to be flipped upside down. And this is so that everything can pair properly in DNA. That's called anti-parallel. And finally, you take that ladder of DNA and we wrap that up into a double helix. So it's twisted up into what looks like a spiral staircase. I really like this image. This kind of pairs 3 very classic images of DNA together. So you have your basic image here that shows you all those nucleotides and how they're structured together. You have a little bit more of a ladder image here where you see the sugar phosphate backbones on either side and the nitrogen bases as the rungs of the ladder in the middle. And then finally you have this classic picture of DNA twisted up in its double helix. Right, now that we've reviewed the structure of DNA, we're going to look at how DNA is replicated. And so this is going to happen always before mitosis and meiosis. You should be studying mitosis right now. And in mitosis, we have to replicate DNA during interphase. So we're going to look at some basic steps of replication. First of all, we're going to start with our DNA molecule right here. And to replicate it, we're going to have to open it up. So we're going to have to split it between the hydrogen bonds. Then we're going to use both of the original strands as a sort of template. And we're going to build in a new strand here on the left and a new strand here on the right. So that we'll finally have 2 completed strands of DNA from our original over here. So we're going to look at that a little bit more in depth. So here we are in the beginning, our original DNA, and we have to split it. And that's done with helicase. This is an enzyme that helps us split the DNA. The next thing we do is we have to add the new nucleotides. Notice that I've got this split strand, this light blue on top and the light blue on the bottom. And we have another enzyme that's going to come in here and it's going to build our new strand. Notice the leading strand is always built in a continuous motion, but if you remember on the lagging strand, this is built in little fragments because we're always building 5' to 3'. Either way, once we've built the left side and we've built the right side, the last thing we have to do, you can see here in this image, we have to twist them back up. So we've opened it, we added our new one in, and now we're twisting it back in with the old. As a result, this is considered semi-conservative because each time we make a new strand of DNA, we use an old strand. Notice here it's in purple, and we are building one new strand in red. So it keeps part old, 50% old, and 50% new, which is considered semi-conservative. So that's DNA structure and DNA replication. Good luck on your air test.